Hello everyone, welcome to my YouTube channel. This is Enoch's tutorials. Don't forget to like, subscribe, and also press the notification bell after watching so you can receive every bit of update from this channel. In this video, we are going to look at regulation of glycolysis, which falls under the concept of integration and control of metabolism. Don't forget to like, subscribe, and also press the notification bell after watching so you can keep on being updated on every update from this channel. Basically, there is a need for us to regulate every metabolic pathway that occurs in the body because overproduction of metabolites can lead to sometimes toxicity. So if the expected amount of every product that you want is achieved, there is a need for us to halt that product or that metabolic pathway. If the amount is also less, there is a need for us to speed up the activity of the enzymes in the pathway so we can get our product at a very faster rate. So we have to regulate the activity of the enzymes in the metabolic pathway so we can tend to either increase the rate of the metabolic pathway or decrease the rate of the metabolic pathway. We're trying to regulate every pathway involves just changing the activities of the enzyme. We have two main ways of controlling the activities of the enzymes in the pathway that helps to either speed up the metabolic pathway or halt the metabolic pathway. We have the cost control method and a fine control method. So the cost control method deals with you trying to change the synthesis or the degradation rate of the enzymes involved in this metabolic pathway. These pathways are speed up by what we call enzymes, and enzymes are proteins which are all synthesized at the genetic level. So if we're able to more of change the activity or change the synthetic rate or the degradation of the enzymes, we're more of trying to halt the reaction or increasing the reaction. So if you're able to change or say decrease the rate of the enzyme synthesis, it means we are trying to more of halt the reaction. If you are increasing the rate of the enzyme synthesis, we are trying to increase the rate of the metabolic pathway. If you more of decrease the rate of the enzymes degradation, we are trying to increase the rate of the reaction. And if we increase the rate of the degradation, we are more of decrease the rate of the metabolic pathway. No, this one is what we call the post synthesis, which is normally controlled at the genetic level. You're trying to control the rate of the enzyme synthesis at the genetic level because enzymes are protein. The next one is what we call the fine control method. The fine control method just deals with the control of the enzymes at a fixed amount. So here we just try to change the activity of the enzyme. So the changing the activity of the enzyme is what we call the fine control method. The first one is just trying to change the rate of its synthesis or degradation at the genetic level. But here we change the activity of the enzyme. So we change the activity of the enzyme by two main processes, which we call the allosterism and covalent modification. We are going to look at how these processes can be used to regulate glycolysis in the body. So allosterism just deals with you using, let's say, allosteric effector or being it an activator or inhibitor to regulate the allosteric enzymes or the regulatory enzymes found at the rate limiting steps of each of the metabolic pathways. And the covalent modification just tries to deal with you trying to introduce a new group or remove a new group to either create or let's say destroy a covalent bond in an enzyme so you can halt or speed up the rate of the reaction. So here you more of create a new covalent bond, here you more of try to inhibit or speed up the rate of the activity of the allosteric enzymes. Now let's come to glycolysis. This one is a summary of the allosteric inhibition of the glycolytic pathway. So with allosteric inhibition, we can have feedback inhibition and feed-forward activation. So the feedback inhibition is when a product tries to inhibit the activity of an enzyme which is at the early stage of the reaction. And feed-forward inhibition is when, let's say, one product at the early stage of the reaction tries to activate one of the enzymes at the later part of the reaction. So here you have the enzymes in each of the metabolic pathways in glycolysis running from glucose 6-phosphate down to pyruvate. Now when we take the whole pathway of glycolysis, there are three main control steps. The first one is conversion of glucose to glucose 6-phosphate by an enzyme hexokinase in the tissues, or let's say glucokinase in the liver. The next one is converting from fructose 6-phosphate to fructose 1,6-bisphosphate, which is catalyzed by an enzyme phosphofructokinase 1 or PFK1. Then the last one is conversion from phosphoenolpyruvate to pyruvate, which is catalyzed by pyruvate kinase. So three main sites are the rate limiting site for glycolysis. Basically, nature has made it such that the first step, which is conversion from glucose to glucose 6-phosphate, is not the control step. This is because the glucose 6-phosphate, which is needed, is also needed in other metabolic pathways. So if this reaction or if this process is halted, it means we can't attain other metabolic pathways. For example, if we have high excess of glucose in the body, we have to convert this glucose to glucose 6-phosphate so it can be converted to glycogen in the liver through glycogenesis. If you are deficit of, let's say, NADPH for fatty acid synthesis or other synthetic mechanism, or let's say ribose sugar for nucleic acid synthesis, you have to convert glucose to glucose 6-phosphate, which enters the PV pathway, which helps in the production of NADPH and ribose sugar for the synthesis of nucleic acids and also for production of more NADPH for other biosynthetic pathways in the body. So glucose 6-phosphate is needed in the pathway. Therefore, the first step is not the rate limiter or the control step in glycolysis. Next is the ninth step or the last step, which is catalyzed by pyruvate kinase. This also helps in control of the whole process because this enzyme can also be inhibited by its own substrate or can be inhibited by some substrate. Now, as soon as we try to inhibit the glycolytic pathway or halt the glycolytic pathway, it means that we have a lot of ATP because glycolysis tends to have produced ATP in the body. So if you have a lot of ATP in the body, it means that it must halt so we can have other glucose being reserved as glycogen. So this one too is not really the control step because if you try to use this step as a control step of glycolysis, we are going to produce some ATP at somewhere step 7. Step 7, that is from 1,3-diphosphoglycerate to 3-phosphoglycerate. Here, ATP is produced through the substrate level phosphorylation. So if you try to halt this pathway, you will still produce ATP in glycolytic pathway, even though we want to stop the pathway. So the last step and the first step is not the control point of glycolysis. Though they can aid, they more of help as the rate limiting step, but they are not the main control steps in glycolysis. The main control step in glycolysis is conversion from fructose 6-phosphate to fructose 1,6-bisphosphate. This control mechanism shown here are what we call the allosteric inhibition. And these enzymes here are what we call allosteric enzymes. They are called allosteric enzymes because they have two sites. One allosteric site, one active site. The allosteric site is a new site on the enzyme that if any compound binds to that site, it more of changes the conformation of the active site so that the substrate can bind. So you're taking this one for instance, the main regulator of hexokinase is glucose 6-phosphate. So this one is an allosteric inhibitor of hexokinase. So if the body want to halt this pathway, if you have a lot of glycogen, a lot of ATP, and a lot of ribose sugars, this one must halt. So glucose 6-phosphate can go and bind the allosteric site of this. As soon as we have a lot of phosphate in the body, it means we are more of deficit of ATP. So phosphate groups can also go and activate. So the activation here is the feed-forward activation, and the backward inhibition is the feedback inhibition. So the plus charge here means activation. So PI can activate hexokinase 1, and glucose 6-phosphate can inhibit hexokinase. In the same way, when we take the last step, when we have a lot of ATP in the body, the ATP can go and inhibit the pyruvate kinase. Pyruvate kinase. And also, when you have a lot of fructose 1,6-bisphosphate, this one is a positive feed-forward allosteric activator of pyruvate kinase. So fructose 1,6-bisphosphate is a positive feed-forward activator of pyruvate kinase. Therefore, when it binds to the active site of pyruvate kinase, it more of makes the active site more regular so that the phosphoenolpyruvate can bind and get converted to pyruvate. So this is a feed-forward activation, and the red ones are the feedback inhibition. The next one is the action of the PFK1, which is the phosphofructokinase 1. We have two analogs of these enzymes. We have one which is PFK2 and one is PFK1. Now for the PFK1 is found only in glycolysis. And the PFK2 we are going to look at in the next slide. The PFK1 is inhibited by high concentration of ATP in the body, high concentration of citrates in the body, sometimes high concentration of hydrogens in the body. If you have high concentration of hydrogen in the body, they can also go and inhibit the PFK1. The PFK1. PFK1 also has its own positive allosteric activator, which we are going to look at in the next slide. So this mechanism, as described here, is the feed-forward inhibition, also the feed-forward activation and feedback inhibition. So the feed-forward activation is when a molecule binds to the allosteric site and make the active site more regular so that the substrate can bind. Then the feedback inhibition is when an inhibitor binds the allosteric site and changes the conformation of the active site so the substrate can bind. So take note of these two. Now the next thing we are going to discuss here is the action of what we call bifunctional enzymes. We said earlier that the PFK1 also has a feed-forward activator. It's called fructose 2,6-bisphosphate. This fructose 2,6-bisphosphate is synthesized by an enzyme called PFK2, which is phosphofructokinase 2. Then this one too is reversed by an enzyme called fructose 2,6-bisphosphatase. These two enzymes, PFK2 and FBPase2, are what we call bifunctional enzymes. They are called bifunctional enzymes because they are found on the same proteins, but they are at different sites. So the same protein has a site for this and a site for this. At the moment this one is active, this one becomes inactive. At the moment this one becomes active, this one becomes inactive. They are found on the same protein and they move of work antagonistically. The function of the PFK2 is to also act on the same fructose 2,6-bisphosphate which is acted on by PFK1. So PFK1 and PFK2 both act on what we call it fructose 6-phosphate. But PFK1, which is in glycolysis, converts fructose 6-phosphate to fructose 1,6-bisphosphate. But PFK2 converts fructose 6-phosphate to fructose 2,6-bisphosphate. The idea here is the fructose 2,6-bisphosphate is a positive allosteric activator of PFK1. Therefore, when you have high concentration of the fructose 2,6-bisphosphate, we are going to have the glycolytic pathway being active because this one will activate the main control enzyme in glycolysis. If PFK2 is active, it can go and convert fructose 6-bisphosphate to more of fructose 2,6-bisphosphate. So the fructose 2,6-bisphosphate will go and activate PFK1 because it is a positive allosteric activator of PFK1. In the same way, it is a negative allosteric inhibitor of this enzyme, which is fructose 1,6-bisphosphatase found in gluconeogenesis. So it will activate this one to speed up glycolysis and inhibit this one to halt gluconeogenesis. So these two enzymes, PFK2 and PF fructose 2,6-bisphosphatase, are what we call bifunctional enzymes. When you have high concentration of this, the fructose 2,6-bisphosphatase, this one will convert the whole of the fructose 2,6-bisphosphate back to fructose 6-phosphate. Therefore, we will not have any of these to inhibit this, to activate this. So it means we are going to halt the glycolytic pathway when there is high concentration of fructose 2,6-bisphosphatase because it will convert fructose 6-phosphate to fructose 6-phosphate. So we don't have any fructose 6-phosphate to go and activate this. So when fructose 2,6-bisphosphatase is in a limited amount, we try to halt glycolysis because it is the positive allosteric effector of the PFK1. In the same way, the fructose 1,6-bisphosphatase is also a positive allosteric effector of pyruvate, as we said earlier, pyruvate kinase. So when this one is inactive, it can convert fructose 6-phosphate to fructose 1,6-bisphosphate to also go and activate pyruvate kinase. So we more halt the glycolytic pathway completely. Understand the concept of the bifunctional enzyme very well, because these ones are what we are going to apply in covalent modification of enzymes. PFK to convert fructose 6-phosphate to fructose 2,6-bisphosphate. which go and activate PFK1. So if PFK2 is inactive, we will not have fructose 2,6-phosphate. Therefore, I will not have the glycolytic pathway being active. Take note, at the moment PFK2 is active, PFK1 is active. But at the moment F2 6-base phosphate is active, it converts all the F2 6-base phosphate to fructose 6-phosphate. Hence, it will halt glycolysis because this one would be inadequate to activate this. If this one is inadequate, it means that we are going to more of allow this enzyme fructose 1,6-base phosphatase in gluconeogenesis to be active. So when we halt glycolysis, we are more going to activate gluconeogenesis. Let's apply this concept in covalent modification of glycolysis. So far, we are done with the allosteric inhibition or allosteric activation. If you have any question, drop it at the comment section and we are going to answer you. Now let's come to covalent modification. This is just a schematic summarized representation of the covalent modification of the enzymes. So, as we discussed earlier, the covalent modification comes from the word that we introduce a new covalent bond in the system, or we remove a covalent bond in the system. So, in enzymes control, the main group which is used in control of enzymes is phosphate group. So here we introduce a new phosphate group, or we cleave a new phosphate group. When we have a lot of glucose in a body, a hormone called insulin is released. When we are deficient of glucose, a hormone called glucagon is released. So let's take this direction one by one and explain what happens when we are, let's say, in a starved state or we are deficient of glucose. When we are deficient of glucose, the pancreatic cells release a hormone called glucagon. And this glucagon binds to glucagon receptors found on the surface of the cell membrane. Now, what happens is when the glucagon binds the cell receptors, there's going to be an activation of an enzyme we call an adenylate cyclase. And the adenylate cyclase does the function of converting the cyclic adenine monophosphate or producing the CAMP, the cyclic adenosine monophosphate, from ATP. When glucagon binds the glucagon receptors on the cell surface, it activates adenylate cyclase, which converts ATP to cyclic AMP. Now, the cyclic AMP is a positive allosteric activator of an enzyme called cyclic AMP dependent protein kinase. So from the word kinase, whenever you hear the word kinase, the kinase just means that it does a phosphorylation from ATP. So when I produce cyclic AMP from ATP by adenylate cyclase, I'm going to activate the cyclic AMP dependent protein kinase. Now the cyclic AMP dependent protein kinase is going to cause phosphorylation of the bifunctional enzyme. Bear in mind, whenever there is phosphorylation of the bifunctional enzyme, as we discussed earlier, the fructose 2,6-base phosphatase is active, but PFK2 is inactive. Whenever we have a phosphorylation of the bifunctional enzyme, we add the phosphate group to that bifunctional enzyme. That's one protein with two active site enzymes. We make the F26-base phosphatase active and make the PFK2 inactive. In other words, when we cause dephosphorylation of the bifunctional enzymes, we make the PFK2 more of active and PFK or F2-base phosphatase inactive. So we said earlier that at any point where PFK2 is active, PFK1 is active. So it means that in glycolysis, when we phosphorylate the bifunctional enzymes, we are more of trying to proceed with the glycolysis. When we dephosphorylate the bifunctional enzymes, we are more going to halt the glycolysis. So let's see how the mechanism goes. When the cyclic AMP activates the cyclic AMP dependent protein kinase, this one causes phosphorylation of the bifunctional enzyme. The phosphorylation comes from removal of phosphate group from ATP. Then it adds that phosphate to the bifunctional enzyme and make the F26-base phosphatase active. Now, when you have F26 bisphosphatase active, it means that, as we discussed earlier, let's go up and see. When you have F26 bisphosphatase being active, it will convert the F26 bisphosphate to fructose 6-phosphate. So we have deficit amount of fructose 2,6 bisphosphate. Hence, there will be no F26 bisphosphate for us to activate PFK1. Hence, glycolysis would halt. So, phosphorylation of the bifunctional enzyme will cause activation of the F26 bisphosphatase, which will eventually halt glycolysis. So let's come here. If we phosphorylate the bifunctional enzyme, we are more of trying to make the F26 bisphosphatase active. The F26 bisphosphatase is going to convert the F26 bisphosphate to fructose 6-phosphate, which is going to make the body be deficient of fructose 6-phosphate. Now, since fructose 6-phosphate is a positive allosteric effector or activator of PFK1, the PFK1 will not be activated when the fructose 6-phosphate is in deficit. Hence, glycolysis will halt. Now, let's come to when we are in a fed state or we have an abundant glucose in the body. If you have an abundant glucose in the body, what happens is insulin is also released from the pancreatic cells. The pancreatic cells. Insulin is released from, I think, the beta pancreatic cells, and the glucagon is from the alpha pancreatic cells. Insulin also binds to the insulin receptors upon its release on the cell membrane, and when insulin binds to its receptors on the membrane, it causes activation of insulin secondary mediators. Now, the insulin secondary mediators does all the work of what we call dephosphorylation. And as we said earlier, dephosphorylation always activates the PFK2. It does so in three main mechanisms. When there is an activation of the insulin secondary mediators, the insulin secondary mediators can first go and activate an enzyme called phosphodiesterase. Then the phosphodiesterase will cleave the cyclic AMP to AMP. So there will be no cyclic AMP to activate the cyclic AMP dependent protein kinase. Hence, therefore, we can't proceed with phosphorylation. Hence, dephosphorylation will occur, which will activate the PFK2. The first mechanism is the activation of the phosphodiesterase by the insulin secondary mediators. And the phosphodiesterase will convert the cyclic AMP to AMP. Hence, there's no cyclic AMP to activate the cyclic AMP dependent protein kinase. Hence, we can't activate the PFK or the F26 bisphosphate. Therefore, the PFK2 is going to be active. That's one. Or the insulin secondary mediators can straightforward go and inhibit the cyclic AMP dependent protein kinase. So we can't proceed with phosphorylation. Therefore, dephosphorylation will occur. The last one is the insulin secondary mediators has a positive allosteric effector or activation on a protein which is called phosphoprotein phosphatase. Now, this phosphoprotein phosphatase is capable of causing dephosphorylation of every phosphorylated protein in the body. So as soon as we activate the phosphoprotein phosphatase, it will cause dephosphorylation of the bifunctional enzyme, and that dephosphorylation will cause activation of the PFK2. And we have PFK2 being synthesized in the body, we are going to have an abundant amount of fructose 2,6 bisphosphate, because the PFK2 will convert the fructose 6-phosphate to fructose 2,6 bisphosphate, as we discussed in the previous slide. If you have dephosphorylation of the bifunctional enzyme, the PFK2 is going to be active. Hence, it will convert fructose 6-phosphate to fructose 2,6 bisphosphate. And this fructose 2,6 bisphosphate will go and positively activate the PFK1, which will proceed with glycolysis. So, in general sense, by covalent modification, phosphorylation of the bifunctional enzymes halts glycolysis, and dephosphorylation of the bifunctional enzymes proceeds glycolysis. Phosphorylation causes activation of the F26 bisphosphatase, which will make the F26 bisphosphate in a limited amount, or convert the F26 bisphosphate to fructose 6-phosphate. Hence, we will not have any F26 bisphosphate to go and activate the PFK1. If you have dephosphorylation, which is catalyzed by release or caused by release of insulin, which can go and activate the phosphoprotein phosphatase, causing this phosphorylation. We are going to more of activate the PFK2. Then the PFK2 will convert the F26 phosphate or F6 phosphate to F26 bisphosphate, and the F26 bisphosphate will go and activate the PFK1. Then the PFK1 can proceed with the glycolysis. So let's check the next slide through and see how best we can use this analogy to explain this. If you have high glucagon level, the high glucagon level activates the protein kinase A. Now the protein kinase A, as we said earlier, is a cyclic AMP dependent protein kinase. So the high glucagon level will cause activation of adenylate cyclase, which will also produce more adenylate cyclic AMP, which will activate this cyclic AMP dependent protein kinase. Now the kinase will cause phosphorylation of the bifunctional enzyme. So it adds a phosphate, or let's say adds a phosphate group to the last serine amino acid of the bifunctional enzyme, and that will make the PFK2 inactive and make what do you call it, the fructose 2,6 bisphosphatase active. These two are the bifunctional enzymes. When fructose 2,6 bisphosphate is active, it will convert the fructose 2,6 phosphate bisphosphate to fructose 6 phosphate. It means at any point where we have more or activation of the fructose 2,6 bisphosphatase, the fructose 2,6 phosphate is going to be in a limited amount. Hence, there will be no this to go and activate PFK1. In other words, when we have high concentration of or activation of the fructose 2,6 bisphosphate, we halt glycolysis. That is for the release of the glucagon. Now, upon release of insulin, when insulin is released, what happens is, to understand this one, it says that glycolysis slows down and gluconeogenesis increases when there's a low fructose 2,6 bisphosphate. So when there's low fructose 2,6 bisphosphate, it means that there is no desk to go and activate positively PFK1. Hence, glycolysis slow and gluconeogenesis speed up. When we are having enough of glucose in the body, or we are in a fed state, insulin is released from the beta pancreatic cells. And as we said earlier, it's go and activate a protein phosphatase, which will cause dephosphorylation of the bifunctional enzyme. And that makes the PFK2 active. When PFK2 is active, it converts fructose 6 phosphate to fructose 2,6 bisphosphate. And fructose 2,6 bisphosphate will more activate the PFK1 in glycolysis. Hence, will speed up or stimulate glycolysis. So glycolysis is stimulated at high fructose 2,6 bisphosphate concentration. High insulin concentration activates protein phosphatase to remove the phosphate from the serine of the bifunctional enzyme, which stabilizes kinase function, which would promote production of fructose 2,6 bisphosphate. So that's the activity of the bifunctional enzyme to control glycolysis through covalent modification. Let's look at the last chart and see if we can also use this one to understand the covalent modification mechanism by the bifunctional enzyme. Having here is the fructose 6 phosphate, which is converted to fructose 1,6 bisphosphate by phosphofructokinase 1 in glycolysis. When we have fructose 6 phosphate, it can be acted on by both PFK2 and PFK1. Now, PFK2 converts the fructose 6 phosphate to fructose 2,6 bisphosphate, which goes and activates what? Phosphofructokinase 1. So, at any point where you have a limited amount of fructose 2,6 bisphosphate, PFK1 is going to be inactive. This enzyme, bear in mind, PFK1 is found in glycolysis, but PFK2 and the phosphatase, the two bifunctional enzymes, are not found in glycolysis. Fructose 2,6 bisphosphatase also converts fructose 6 phosphate, or let's say fructose 2,6 bisphosphate, to fructose 6 phosphate, making the fructose 2,6 bisphosphate at a limited amount. So at any limited amount, we are going to halt glycolysis. In other words, when we have glucagon being released, glucagon releases causes low concentration of fructose 2,6 bisphosphate. Whenever glucagon is released, it causes low concentration of fructose 2,6 bisphosphate. This is due to activation of the fructose 2,6 bisphosphatase, which converts the fructose 2,6 bisphosphate to fructose 6-phosphate. When you have low concentration of fructose 2,6 bisphosphate, glycolysis slows down and gluconeogenesis speeds up. At any point we release glucagon, we more of activate the cyclic AMP dependent protein kinase, and this one causes activation, or let's say phosphorylation, of the bifunctional enzyme. And when we phosphorylate bifunctional enzyme, as seen here, we are going to make the fructose 2,6 bisphosphate phosphatase active and fructose 2 PFK2 inactive. When we release insulin, insulin activates the phosphoprotein phosphatase, which causes dephosphorylation of the bifunctional enzyme, then activate PFK2 and inactivate fructose 2,6 bisphosphatase. Whenever PFK2 is active, we are going to have high concentration of fructose 2,6 bisphosphate. So here, when you have high concentration, or let's say PFK2 being active, we are going to have high concentration of fructose 2,6 bisphosphate. Glycolysis will speed up and gluconeogenesis will limit or go down. Thanks for watching. Don't forget to subscribe to watching so you can receive every update from this channel. Thank you.